Hi everyone, I would like Biochemical Society and Portland Press are very pleased to welcome you all to this webinar, which is part of our Biochemistry Focus Early Career Research webinar seminar series. So the topics in this series will include different research topics in the molecular biosciences, as well as practical sessions to support career development. So each webinar will give you the opportunity to ask questions via text, and we also welcome suggestions for future topics and speakers in the future for these webinar seminars. So please check the website to check for more details on these exciting opportunities. So my name is Gloria Lopez-Castejon, and I am a Sir Henry Dale Research Fellow at the University of Manchester in the Lydia Becker Institute of Immunology and Inflammation, and will be chairing this webinar today. So in fact, this is the first of our early career research webinars. So we are delighted to give the opportunity to Jack Green and Cian, who are both early career researchers, to share the work with the bioscience community. And first of all, I would like to change to Dr. Dominika Guska, that will tell us a little bit more about these new early career research webinar seminars. I think that was a great video. So Dominika already mentioned the webinar title. So for today, we'll be hearing about inflammasomes and the plant resistosome. So I'm also very interested in inflammasomes, so I think it's a very exciting seminar today. And we will be hearing about inflammasomes. And for those that don't know, inflammasomes are molecular complexes which are essential for inflammatory responses and basically the response to damage in your body. So they are very important not only to keep you healthy because they have to function properly, but also because their dysregulation is being linked to disease and conditions such as, for example, atherosclerosis or Alzheimer. So it is a very attractive therapeutic target, and we need to know better how it works. The plant resistosome and inflammasome actually share very important similarities, and it also shares highlights the importance of studying different organisms and different kingdoms in order to better understand the inflammatory process. In fact, discoveries of inflammasome precursors, or the NLRs, in plants have been essential to gain a better understanding on the inflammasome world and inflammation. So hopefully today we'll also hear about the NLRs and how the plant NLRs cooperate in pairs as networks, with one receptors acting as sensors and others helpers, and contributed to immune responses at programmed cell death. So we'll hear more about this in a second, but I will remind everyone that you can ask questions via the chat and the text. So please, as we go along, if you have any questions, just type them there. We will answer the questions after Jack and Cian have done their presentations. So our first invited speaker today is Jack Green, and Jack is a presidential fellow at the University of Manchester. And he completed his PhD at the University of Sheffield, and then he joined the University of Manchester as a postdoctoral researcher. So he's very interested in understanding the mechanisms that regulate the activation of inflammasomes, and in particular, the NLRP3 inflammasome. He's done many discoveries in this area, and he was awarded a presidential fellowship at the beginning of this year. And he will further explore inflammasome in these opportunities it has to establish his own research and his independent career. So Jack will be talking today about how ion fluxes regulate inflammation and the role of chloride in NLRP3 inflammasome activation. So over to you, Jack.
Thank you, Gloria. Can you see this all right?
Yeah.
Yeah. Okay, thank you. So hello, everyone. I'd first off like to thank the Biochemical Society and its organisers for giving me the opportunity to present my research to you today as part of this early career research webinar series. So as Gloria said, my name is Jack Green. I'm a Presidential Fellow at the University of Manchester, and I'm going to be talking to you today about some of the research that I've been doing over the past few years, looking at the role of chloride in the activation of the NLRP3 inflammasome. So inflammasomes are key components of the innate immune system, which work by promoting inflammation in response to infection and tissue damage. So there are several different types of inflammasome, which vary largely by the different stimulus that they are activated by. So here are just a few different types of inflammasome receptors, but there's a whole variety of them expressed in cells. And upon activation by their particular stimulus, you get the formation of the inflammasome complex, which consists of the inflammasome receptor, the scaffolding protein ASC, which then oligomerizes into a large cytosolic speck, on which the protease caspase 1 binds to and is activated, leading to the processing of the pro-inflammatory cytokines IL-1 beta and IL-18, as well as the pore-forming protein gasdermin D. And this results in a very inflammatory form of cell death called pyroptosis, and the release of cytokines into the external environment, which then helps propagate the immune response. So like I said, there's a variety of different types of inflammasomes, but the one that I've been focused in the last few years is the NLRP3 inflammasome. So the NLRP3 inflammasome is of particular interest as numerous studies have shown that it contributes to the worsening of several different sterile inflammatory diseases that are very prevalent in our society. And unlike other inflammasome sensors, NLRP3 can be activated by a wide range of diverse stimuli, allowing it to be activated in a range of different scenarios. And so different types of pathogen-associated molecular patterns, PAMPs, or damage-associated molecular patterns, DAMPs, can lead to the activation of the NLRP3 inflammasome, resulting in an increased processing and release of the inflammatory cytokine IL-1 beta, which can then have effects throughout the entire body, and has been implicated in the worsening of Alzheimer's disease, atherosclerosis, rheumatoid arthritis, diabetes, and many, many other diseases that we don't really quite have a good clinical therapy for. And seeing as NLRP3 has been implicated in the worsening of these diseases, if we can understand the mechanisms surrounding NLRP3 regulation, this could help us develop effective therapeutics against these disorders. And as such, there's actually been quite a lot of intense research into the NLRP3 inflammasome, and we know quite a bit about mechanisms surrounding its activation. And it's typically considered to be a two-step process, where we first have a priming signal, which is driven through receptors such as TLR4, where we get activation of NF-kappa B, leading to the transcription of IL-1 beta and NLRP3 itself. Following this, we get an activation signal, which is largely considered now to be driven through the efflux of potassium from the cell. And this has been achieved through a variety of different mechanisms. It can be driven through membrane receptors, pore-forming toxins from bacteria, or particulates damaging the cellular membrane. And then this leads to the formation of the inflammasome complex and the processing of cytokines and pyroptosis. So although potassium efflux has been well described as an activator of the inflammasome, increasing evidence over the last few years has suggested that there may also be a role for chloride efflux in regulating this response. And so here are a few papers that have come out over the last few years. I'm afraid I don't have enough time to go into them in a lot of detail, but if you're interested, there's some papers that you can read. But a lot of evidence suggests that chloride efflux can be implicated in NLRP3 activation. And for the purposes of this presentation, the main points I want to make from these papers is that if chloride ion flux has been found to modulate NLRP3 activation in vitro, And several diverse chloride channel inhibitors have been shown to be effective at blocking NLRP3 activation. And therefore, this suggests that chloride channels could be involved in activation of the NLRP3 inflammasome. And so if we can identify which channels are implicated, then this could help direct which therapeutics we can develop to target NLRP3 mediated diseases. And so this led us to the hypothesis that one particular chloride channel, the volume regulated anion channel, could be one of these channels upstream of NLRP3. And as its name suggests, VRAC is primarily involved in the control of the cell volume of the cell. So most cells express VRAC. At baseline, it is closed, but if the cell finds itself in hypertonic environment, water then floods into the cell down its osmotic gradient, causing it to swell. And to prevent it from bursting, VRAC is activated on the plasma membrane, resulting in chloride efflux from the cell, and then water follows that, causing the cell to shrink back down to normal volume, and VRAC closes itself. So what has led us to propose that this channel could be involved in NLRP3 activation? One, well, chloride channel inhibitors that have been reported to block NLRP3 activation have also been reported to be VRAC inhibitors. And secondly, this process of hyperosmotic shock has been shown to be sufficient to activate the NLRP3 inflammasome in macrophages. So this has led us to the hypothesis that VRAC may be an essential regulator for NLRP3 activation. And we decided to test this hypothesis using a range of pharmacological and genetic approaches. And this, the results of this study have just been accepted in eLife, so you should be able to read the full story very soon, but I'm just going to go over some of the highlights of this, this study for you today. But before I get onto that, I would like to talk a little bit more about VRAC. So the molecular identity of VRAC was identified 5 or 6 years ago now, and it is now known that it's a hexameric channel composed of this LRRC8 family of proteins. And there are 5 flavors of this LRC8A protein. There are A to E, and they can come together in a range of different combinations as long as you have at least one of the LRC8A family members in the channel. And following this identification, there's been a lot of interest in looking at these channels, including the generation of the cryo-EM structures of VRAC and in the presence of an established VRAC inhibitor, DCPIB. So you can see here this top view of the channel. Each of these different colors is a different subunit, so there's 6, and then there's the central pore region here. And this established VRAC inhibitor has been proposed to bind to this like right in the middle of the pore. So this was very interesting information for us because we were able to use this structure to guide our pharmacological approach to test our hypothesis whether VRAC was involved in NLRP3. So we took this crystal structure and we computationally modeled other potential VRAC inhibitors and to see how they interacted with VRAC. So this is just a zoom in here of the pore structure again. So this is the central pore. And then these are the 6 subunits surrounding the pore. And then this is the cryo-EM pose of DCPIB blocking this central pore. And we then computationally modeled in flufenamic acid, mono, NS3728, sulfonic calixarine, and tamoxifen. And we showed that they showed a preferential binding to the central pore region, much like DCPIB did. And so this allowed us to use this panel of drugs as predictive VRAC inhibitors to see what impact they have on the inflammasome response. But before we got to there, we wanted to first test our hypothesis that these were effective VRAC inhibitors. And to do this, we initially did a VRAC assay in HeLa cells, where we made them express a mutated version of YFP. So this is a yellow fluorescent protein that contains mutations that make it sensitive to halides, such as iodide. So in the presence of iodide, the fluorescence of the protein is quenched. And so iodide normally cannot enter the cell unless you have open chloride channels on the plasma membrane. So if we perform these, if we add iodide to the cell in the presence of hypertonicity, where we have open VRAC channels, you can see here that we get this, this quenching response occurring. And then we can test the ability of VRAC inhibitors to block this quenching response. So these are some videos when we add the iodide, you can see this quenching response occurring. If we quantify this over time, you can see at baseline that there's no difference. And then when we add the iodide, you see this rapid quenching. So the difference between these two lines shows the open chloride channels at the point of iodide addition. And so if we repeat this experiment with the established VRAC inhibitor DCPIB, this response is completely blocked. So this shows that this difference in response is due to VRAC. So what we did is we put in our panel of potential VRAC inhibitors into this assay, and you can see in the vehicle we had this quenching response, and this response was completely blocked by tamoxifen, mono, DCPIB, flufenamic acid, and NS3728, but not by calixarine. So it appears that all of these compounds, apart from calixarine, are effective VRAC inhibitors. But we were wanting to look at inflammasomes in macrophages, and that was a HeLa cell, so we wanted some readout of its ability to block VRAC in macrophage cell type. And as I said before, VRAC has been involved in the regulation of cell volume in response to hypertonicity, so we decided to look at cell size in response to hypertonicity. So we took a macrophage, you put on a hypertonic shock, the water floods in, and then VRAC is activated to cause water to leave the cell, causing the cell to shrink down in a process known as regulatory volume decrease. And then when we do these experiments, so when we add hypertonicity to a macrophage, you see this rapid and acute increase in cell size, which then subsides over time as the cell restores its volume. And when we do this with a VRAC inhibitor, this is completely absent, showing that this is what happens when we block VRAC. The cell can no longer control its volume. And again, we put all of our compounds through this assay again, and we found that in complete matching with the experiments we did in the HeLa cells, that all of the inhibitors were unable to block this reduction in cell volume apart from calixarine. So we were pretty confident we had a panel of drugs that contained quite a few good VRAC inhibitors that we could test their effects on the NLRP3 inflammasome. So then we've moved on to quite a traditional method to activate the NLRP3 inflammasome, where we took bone marrow-derived macrophages from mice, we LPS primed them, and then we incubated them with a VRAC inhibitor at the same dose that we used in the previous experiments to block VRAC, and then activated the inflammasome with ATP. And so there's quite a few different readouts for NLRP3 inflammasome activation. And we first off looked for the presence of mature IL-1 beta in the supernatant of cells. And as you can see here, when we add ATP to the cells, we see quite a lot of IL-1 beta starting to be released from these cells. And interestingly, despite the fact that we found that in the VRAC assays that all of them but calixarine were effective VRAC inhibitors, we saw a differential effect where only mono, flufenamic acid, and NS3728 were able to block NLRP3 inflammasome activation. This was consistent amongst other readouts of inflammasome activation, so we could look for the formation of these ASC oligomers inside the cell by cross-linking the insoluble fraction and blotting for ASC. So you can see these multimers here, the cleavage of gasdermin D, the cleavage of caspase 1, and the cleavage of IL-1 beta. And in every case, it's the same mono, flufenamic acid, and NS3728 were the only VRAC inhibitors that could consistently inhibit the inflammasome. And so MCC950 and MBC here are NLRP3 inhibitors to show that this was an NLRP3 specific assay. So this led to the question that if all these inhibitors are working differentially, are these inhibitors actually targeting VRAC to inhibit NLRP3? So we then decided to go along the genetic approach, and we decided to genetically remove VRAC from these cells. And so to do that, we decided to knock out the major protein that was essential for functional VRAC channels in cells, so this LRRC8A. So global LRRC8A mice are lethal, so we used Crelox to generate a myeloid-specific knockout mouse line. So we floxed the major coding exon of LRC8A and then crossed this with a CX3CR1 Cre mouse to mediate the deletion here. And then once we did this, we generated mice that will legitimate wild type or macrophage-specific VRAC knockout mice. And then when we isolate macrophages from these cells, we can see that the knockout mice have completely lost their expression of LRC8A. And so the first thing we tested in these animals is their ability to control their volume, as this is the most common phenotype reported in other cell lines. So it would tell us whether we had a functional knockout in these animals. And so we took macrophages from wild type and knockout mice and did our RVD assay. And so in these time lapses here, you can see in the wild types, once we add the water, they swell up and then quite quickly return back down to normal cell volume. Whereas you see quite a striking phenotype in the knockout where they swell up and they stay swollen indefinitely, hours at a time. They don't lyse, they just stay swollen. It's a really interesting phenotype. And when we quantify this again, you can see that these knockout cells are unable to now control their volume. So the macrophages from these mice appear to have a functional phenotype, and so we should test them for their ability to regulate the NLRP3 inflammasome. And so we went back to do our inflammasome model. So we took wild type and knockout bone marrow derived macrophages, LPS primed them, and then activated them with a range of NLRP3 stimuli. And interestingly, probably not surprisingly based on the drug data, we found that in the knockout mouse macrophages, there was no defect in IL-1 beta release in response to ATP, the pore-forming toxin nigericin, or the particulate silica and imiquimod. And then the next thing we wanted to test, because we saw no defect here, was whether the VRAC inhibitors would still be effective in these cells when they don't have VRAC for them to target. So is VRAC important for this inhibition or not? So again, we did the same experiment, but we added the VRAC inhibitor in. And matching this experiment, we saw in the wild type and knockouts, there was no defect at baseline. But interestingly, the VRAC inhibitors, flufenamic acid and NS3728, were equally as effective at inhibiting NLRP3. And to finally examine this, we decided to go in vivo, and we did a mouse model of peritonitis, which is NLRP3 dependent, where we inject a low dose of LPS into the peritoneum. 4 hours later, inject a high dose of ATP, and then look for IL-1 beta in the peritoneal cavity. And in wild type mice, when we do this, you can see this induction of IL-1 beta in the peritoneum, which is blocked by the VRAC inhibitor and NLRP3 inhibitor. And when we did this in the VRAC knockout mice, there was no difference in the vehicle, and this was still inhibited by the VRAC inhibitor. So all of this evidence together suggests that VRAC is completely dispensable for DAMP-induced NLRP3 activation and is not essential at all. But one last thing I'd like to just point out. So I said at the beginning of the talk that hypertonicity alone has been shown to be an activator of the NLRP3 inflammasome. And since we had shown that these cells could no longer control their volume to hypertonicity, we wanted to see what effect the knockout had on the inflammasome activation in these contexts. So we LPS primed wild type and knockout bone marrow derived macrophages and subjected them to hypertonicity. And interestingly, despite being able to still respond to DAMP-induced NLRP3 activation by ATP, the hypertonicity-induced inflammasome response was completely lost in the knockout animals. And we further confirmed this by Western blotting. So you can see the cleavage of IL-1 beta and caspase-1 in the wild type here, blocked by the NLRP3 inhibitor MCC950, and is completely absent in the VRAC knockout mice, whilst ATP is maintained. So this suggests that VRAC is actually essential for hypertonicity-induced NLRP3 inflammasome activation. So I'd just like to summarize what this study has shown to you now here. So this suggests that DAMPs that are sufficient at activating the NLRP3 inflammasome act predominantly via other chloride channels. We can't disprove that they don't work via VRAC, but at the very least, VRAC is redundant. It can be compensated for by other channels. Whereas hypertonicity, which is unable to activate other chloride channels, exclusively works on VRAC. And putting further evidence on the fact that chloride channels are important for the activation of the NLRP3 inflammasome. And that VRAC inhibitors, although being able to block VRAC, are very likely to be able to block other chloride channels that contribute towards this response. We, however, cannot disclude that VRAC inhibitors may be working at some other downstream part of the activation, but this requires further research. So all that's left is to thank you all for listening and to acknowledge a lot of people that helped with these studies. So there's Tessa, there's Lucy, there's Arthur that have all helped with carrying on of this project. And I'd like to thank Professor David Brough. So all of these experiments were done during my postdoc in his lab. So I'd like to thank him for his advice during this time. So yeah, thank you very much.
Great talk. Thanks, Jack. I don't know you heard before. That's a great talk, so thank you very much. So we'll now move on to the next talk, but I'd like to remind you that you have any questions for Jack, and now maybe the seminar is fresh in your memory, you can type them in the question box. So I'm quite glad to introduce you to Cian Dugan, which is the second invited speaker for today. And he's from Tolga Bozkurt, sorry for the pronunciation, lab at Imperial College London. And Cian studies the immune responses of plants to pathogens. As I said before, this is very interesting to really understand how plants respond to pathogens and the role of NLRs. And his work is focusing on how the NLR from plants work during infection, in particular during the Irish potato famine pathogen infection. So the talk of Cian is called the plant resistosome, an ancient cell-intrinsic self-destructive machinery to eliminate pathogen spread. So over to you, Cian.
Hello, and hopefully you can see my slides.
Yeah, that's great, thank you.
Great, so thank you first of all to the Biochemical Society for this opportunity. It's great to be here. So as you said, I'm presenting the plant resistosome today. My work focuses on plant NLRs, their localization, and the plant resistosome. And this is important because in order to have disease-resistant crops and to feed the planet, we need a robust immune system. But further work needs to be done because evolution of the pathogens is overcoming our crops' immune systems. So understanding this further will enable us to breed and engineer disease-resistant crops. As I should have said, I'm from the Bozkurt lab at Imperial College London. And in this lab, we study the potato blight pathogen, Phytophthora infestans. It's a fungal-like pathogen, although it's phylogenetically unrelated. It infects plants when when a spore lands on the leaf surface or another surface of the plant and penetrates into the tissue. Inside the tissue, it forms hyphae between the cells. And then it uses these hyphae to poke into the plant cells, forming finger-like appendages called haustoria. This is what it looks like in a confocal microscope. So in red here, you have the plasma membrane, and in green here, you have a membrane surrounding haustoria around these finger-like appendages. This is a plant membrane, but it surrounds the pathogen. Across this membrane, the pathogen injects effector proteins. These are virulence factors, and their job is to subvert the plant immune system. But plants often have NLRs, sensors to detect these effector proteins. And when this happens, they trigger programmed cell death. And this is what it looks like in the context of the leaf. And the job of it is to sacrifice a few plant cells in order to prevent the pathogen from spreading. NLRs in plants are multi-domain immune receptors. If we start at the C-terminus, here we have the LRR domain. Its job is to detect the pathogen effectors. In the center, we've got the MB-ARC domain. It provides nucleotide binding and exchange. So this enables the off-on switch of this protein. And at the N-terminus, we have a variable domain. Generally speaking, it's a cold-cold domain or a tier domain. And the job of this is to provide downstream signaling or even more directly, program cell death. So in terms of understanding these NLRs, it's best to think about an evolutionary perspective. We think that they started as ancestral multifunctional NLRs. whereby the NLR was capable of detecting the effector and triggering programmed cell death and disease resistance. Some have stayed that way, including ZAR1, which I'll mention in a minute, but others have split their functions over sensor-helper pairs, whereby the sensor detects the effector protein and the helper is required for triggering programmed cell death and disease resistance. This split function has enabled the sensors to diversify, and even for the evolution of disease-resistant networks like this. This is the NRC network. It is a superfamily of cold coil NLRs, which detect a wide variety of pathogens at the top here via their effector proteins. It uses a wide variety of sensor NLRs to do so, and a smaller number of helper NLRs, which are related. We focus in our lab on NRC4, as it is an immune hub for resistance to potato blight, as you can see here. Our understanding of this NLR, this cold coil NLR, has really improved recently, thanks to the first full-length structure of a plant NLR, ZAR1, the cryo-EM structure of it. And what was found was that upon activation, ZAR1 oligomerizes into a pentamer, this resistosome, and this coincides with programmed cell death. As I mentioned, it's called a resistosome, but it's not the only resistosome. Even more recently, a tetrameric resistosome was found from a tier NLR. However, cold coil NLRs are related to our research, so I'll focus on that further. So with ZAR1, the model is that it is in an inactive state, tightly bound around ADP. Upon activation through the effector protein, the ADP is exchanged for ATP or DATP. This enables a massive conformational switch whereby the first alpha helix of the cold coil, shown here in beige, flips out, and then you have the oligomerization into a pentamer. So each of these 5 copies of ZAR1 have their alpha helix sticking out, and they form a funnel-like structure. This coincides with the association of the pentameric resistosome with a membrane. So the model is that this NLR sticks into the membrane with the alpha 1 helix, and this enables programmed cell death. However, we don't know how this membrane association leads to programmed cell death. It could be via a pore formation, but we also don't know which membrane it localizes to. I should say, though, that this is a massive breakthrough in the field because these NLRs, we don't have any downstream components that are known for their cell death. So it is quite likely that it is the elicitor of the cell death itself. So in order to understand this further, we are really interested in the cold coil domain because we know several papers had been published showing that actually the cold coil domain was sometimes sufficient to trigger cell death, but we wanted to study this in an unbiased way. So a team led by Dr. Hiroaki Adachi at Commune Lab in Norwich, our collaborators, and they made a truncation library of NRC4, this cold coil immune receptor we study. They took a autoactive mutant of it, whereby it switched always on with ATP, and they sequentially and randomly truncated it from the C terminus to the N terminus. In this figure here, orange shows the presence of cell death and blue shows the absence of cell death. So you can see that the full-length NLR at the top gives programmed cell death, but as soon as they made the truncations, they lost programmed cell death, which was a failure until they noticed one very small construct, 29 amino acids, was giving some cell death. This was striking. So both our lab and their lab repeated these experiments with a tag YFP So 29 amino acids fused to YFP, and we found that this gave cell death, weaker than the full length, but it gave cell death compared to the empty vector. And we wondered if this cell death was facilitated by some oligomerization like a resistosome. So I investigated this with confocal microscopy and found that the 29 amino acids from the very N-terminus of NRC4 fused to YFP formed puncta in the plant cell. Some of these associated with the membrane, and we hypothesized that perhaps the YFP was acting as a scaffold and mimicking the resistosome formation. This made sense to us because we had read that these fluorescent proteins were capable of forming a wide variety of oligomers, including pentamers like the resistosome. So we made a mutant of YFP that blocks oligomerization, making it monomeric, and this one here. And we found that it lost the puncta formation and diminished the cell death. So this suggested that the YFP was really acting as a scaffold. We next made a mutation in the 9th position from leucine to glutamic acid, excuse me, creating negative charges there. And this actually also abolished the puncta formation and the programmed cell death. I'll refer to this later as the cell death suppressor mutant. And so clearly the 29 amino acids and the YFP are together required for puncta formation and cell death. So this N-terminus is a cell death region for sure. So we compared, or Hiraki and Joe Wynn compared NRC4 and ZAR1's N-termini and found that they were remarkably well conserved. They then went on to see if there was conservation throughout NLRs in plants, and they found that the cold causamine NLRs, 20% of them had a motif in this region, which we nicknamed the MADO motif because of the beginning of its consensus sequence. So it wasn't just conserved in terms of sequence, it was actually functionally conserved, because if you take ZAR1, ZAR1's N-terminus and you put it onto NRC4's N-terminus, it retains its cell death capabilities and its resistance capabilities. So here you have the chimera giving cell death compared to the wild type, and there's no cell death in the negative control. This led us to suggest the following model based on ZAR1, that upon activation, the NLR's first alpha helix flips out, and the NLR then forms a pentameric resistosome and maybe even inserts into the membrane, triggering programmed cell death. But which membrane? We still don't know. So Tolga and I had looked at the NLR with confocal microscopy in live cells of the plant. And we found that when it's fused to GFP, you can see that it's in the cytoplasm on the periphery of the plant cell. For anyone who's unaware, the red I've labeled here is the massive central vacuole of the plant, which pushes the cytoplasm to the extremes. Upon infection, however, it really strikingly changes. So NRC4 GFP accumulates at the host pathogen interface, this membrane surrounding the pathogen. So these finger-like appendages, we know, are really critical to the pathogen because this is where they secrete the effector proteins, the virulence factors. We also know that some of these effector proteins are recognized by NRC4, and they even localize to that membrane. So this really was mind-blowing to us. So it suggests to us that this is the site for effector recognition. It suggests that it's where immune activation is taking place and possibly even cell death execution. But first, do other NLRs localize here? Is this just what all NLRs do? The answer is no. ZAR1, for instance, doesn't accumulate at the host pathogen interface. It's in the cytoplasm here, whereas NRC4 accumulates at this region. So in order to understand this more, we wanted to know what region of NRC4 makes it special. Why can NRC4 do it, but ZAR1 can't? Well, we thought the obvious place to start was the N-terminus, because the ZAR1 paper suggested that the very N-terminus, this alpha-1 helix, was facilitating the membrane association. That's what their data said. So we made that chimera swap, as I mentioned earlier, and it did not alter NRC4's localization. It still accumulated. We made a reciprocal swap, and ZAR1 didn't gain the localization that that NRC4 has. So this region is clearly important for immunity and cell death, but it doesn't determine the accumulation of the NLR NRC4 to the host pathogen interface. We've done further studies on this, and it turns out that the other parts of the core codomain are definitely crucial to the accumulation of the host pathogen interface. So the fact that this was a region for cell death, but not a region for the accumulation phenotype, meant that we can mutate it and disarming it from triggering cell death, but not lose the accumulation, which we confirmed here. We lose the cell death compared to the wild type, but we don't lose the accumulation. This, this is what we call the cell death suppressor mutant that I mentioned earlier. So we use this mutant to study NRC4 further in the active and inactive scenarios. So here we have the inactive scenario where NRC4 is in the cytoplasm. Hopefully you can see that it's colocalizing with the cytoplasmic BFP marker here, and less so with the membrane marker, which is in pink. I should say that these images are taken at the bottom of the cell, so it's curved. That's why it looks a bit weird. We confirmed these observations with membrane enrichment experiments, and found that NRC4 mostly associates with this soluble fraction, the cytoplasmic fraction, when it is inactive, less so with the membrane fraction on the right there. Apologies if you can't see my cursor. There we go. When we introduce a second mutation to this protein, switching it into the always-on situation, the active situation, it won't give cell death because of this, because of the L9E suppressor mutant, but it will activate it. So we believe this will form the resistosome. And actually, what we saw was puncta throughout the plant cell, which was really remarkable. Now these puncta are associating with the membrane area. It's on top of the pink signal more so than the inactive scenario. And we confirmed this observation with the membrane for, enrichment experiment again. And we found, you can see that NRC4 is less in the cytoplasm compared to the membrane. It's more in the membrane than the cytoplasm. And what was really remarkable was that actually there was this high molecular weight band here, which could be SDS detergent resistant oligomers, such as resistosomes. So this really excited us. And in order so the next thing we did was we took these same mutants and we reintroduced the pathogen, because these were done in the absence of the pathogen. And I mentioned earlier already what happened with the inactive scenario. This mutant still accumulates at the host pathogen interface. What we then did was we took the autoactive suppressed mutant, and we found that actually it doesn't accumulate anymore. It was at the plasma membrane and the membrane surrounding the pathogen. So it doesn't accumulate, which gives us a unique ability to dissect the function of this accumulation phenotype. And some data I won't go into here because I don't have time, suggests that this accumulation phenotype is really for immunity to the pathogen, because when we remove this suppressor mutant, the autoactive NLR gives cell death but does not give resistance. So this is, we think, because the autoactive NLR isn't accumulating at the host pathogen interface. So in summary, NRC4, we believe, provides resistance by accumulating at the host pathogen interface. It does this when an effector protein or effector proteins are secreted by the pathogen. many of these localizing to the membrane surrounding the pathogen. NRC4 is then deployed by the plant in the inactive form. It activates when it detects the effector protein and forms the resistosome. This resistosome triggers programmed cell death and disease resistance. And I'd like to thank everybody here, including our collaborators in Norwich, my group leader, Toga Boskert, and everybody else in the lab, and Imperial for funding me during my PhD. And I will go on to take questions later. Is anyone listening to me?
Thanks a lot. Yeah, everyone was. It was a great presentation. Thank you very much. And we could see your cursor, so that the arrow, so that was good. So I would like to encourage people, if you have any questions, please type them in the question box, and I will be reading them as they come, so we can start with our discussion. So let me see if we have any questions. Same way, while we wait for some, it always takes a bit of time for people to type the questions. So I might start asking one. I'll start with Jack, because he did it first. And I always find with the LC's LRC8, like, I guess the key question is, why is just hypotonicity being the one that mediates that chloride channel? Or, you know, why does ATP and all the others don't, and just what does it make it special?
So the way I kind of interpret it is that hypertonicity is a very specific stimulus. So it's unable to activate other chloride channels. It's only able to activate VRAC because of the mechanism of action of it. Whereas other DAMPs might be activating different chloride channels to achieve different effects. I think an area of interesting research that we can go down is it just hypertonicity that goes through VRAC? So there are other ways of activating VRAC rather than hypertonicity. So we're going to need to study whether these are also able to activate the inflammasome via a similar pathway.
Okay, thank you very much. And then I just wanted to ask if you've checked that in the knockouts, their compensation from other chloride channels. So is, for example, the clicks, are they more overexpressed, or are they more functional, or could they compensate that way?
So we've looked at expression of these proteins by RNA, and there doesn't seem to be any at baseline, at least, any real change in expression of any components. But what is interesting, I didn't have time to go into it, but it's in the paper. But when we do the volume regulation and we put on VRAC inhibitors on top of that, in the knockouts, there's an enhanced. They swell even more, and then they burst.
Oh.
So there is something going on that's still activated in these conditions, but we don't know what yet.
Okay. So there's a question also following on that. The question is whether you think that VRAC inhibitors may be inhibiting the gasdermin D pore.
That's that's quite interesting. It could be, but I mean, it's at very least inhibiting upstream of gasdermin D pore as well, because when we look inside the cell, we're not seeing any cleavage of IL-1 or caspase or gasdermin, so it's blocking something upstream. We've not looked after activation to see whether it can still block the pore as well, but there is papers out there suggesting that chloride channels are also involved in the gasdermin pore formation in the membrane. I think Carl Kunzelman has been involved in that.
Okay, thank you very much. So there's some questions for Cian now. So one of the questions is they're asking why do only 20% of the CCNLRs have the MADA motif?
Yeah, that's a really good question, actually. So what we found was really interesting. So the NLRs that had the MADA motif were the ones that are known or thought to be helper NLRs or multifunctional NLRs. So basically, the ones that we think trigger the programmed cell death have the MADA motif, whereas the ones that don't have it are the ones that detect the pathogen effectors. So it's through the evolutionary model, use it or lose it. If you don't need the MADA motif, they just mutated it. It didn't matter. They're not triggering programmed cell death. They're sensing the pathogen effectors.
Mm-hmm. Okay. And they've also asking whether it has been checked whether the sorry, I was going to say NLRC4. Sorry about that if I say
yes,
NRC4.
No relation. No relation.
Yeah, I know. Whether it self-associates and whether it's active. And sorry, I'll read it again. Have it been checked if NRC4 dimerizes self-associates and also the active and the inactive forms? I guess if they form dimers or they associate to each other. If I understood correctly.
So it's a little bit tricky to do certain experiments with the plant NLRs because they're very low expression. But what we can say is when you do a co-IP with two different tagged NLRs, NRC4, you can pull down itself. So they definitely associate with each other, probably directly interacting. There is also some other evidence from our collaborators that they form dimers. But when they're inactive, but in terms of we've had trouble just with the actual assay of doing like blue native page. It's a very challenging assay to show that they form oligomers in native conditions.
I think I'm gonna jump in just to have a question. When you said that higher band might be a potentially oligomer or something, I wonder whether you've also thought whether it could be a post-translational modification potentially instead of an oligomer.
Yeah, I think it's probably I mean, I guess it could be. I would have to. It's quite large, and the other thing is that we're talking about. It would have to happen quite quickly, I'd imagine, for it to be a post-translational modification. But yeah, I think that's something worth considering. It doesn't. Let me just be clear though. It doesn't come with the inactive form. So it's only when it's activated it has it. So it's possible, or yeah, we can investigate that as well.
So other question for that says, is it possible that a sensor NLR partner is localizing NRC4 to the host area?
Can you say that again? Sorry.
I think you might have touched on that already. Is it possible that a sensor NLR partner is localizing an NRC4 to the, I don't know how you pronounce that, host area?
Host area, yeah. Actually, it seems like that's not the case because we have investigated the localization of the sensor NLRs, and so far it seems like they do not accumulate. So we don't think the sensor NLR would be carrying them. There could be another NLR involved for sure, but probably not the sensors. I should mention as well, though, that the sensor NLRs often detect effector proteins indirectly. So it's not a big surprise that they don't accumulate at the host pathogen interface.
Okay. So another one, say, what happens if you co-express NRC4 alleles with two different tags? Do they co-IP in the case of the MHD mutant, but not if you use the wild types?
So if you pull down the two different tagged NRC4s, if you have the yes. So when you have the autoactive NLR, that is a good question. I actually haven't done that experiment yet. It's something that I was about to do before lockdown and haven't done, but I would say yes. However, there's one thing that is a bit of an issue is when you have activated when you have oligomers and when you also have membrane binding proteins, you can lose them in the extraction process, which I'm sure plenty of people here are familiar with. So I will do those experiments, but I'm expecting them to be not as straightforward as I'd hope.
Yeah. And there's another question that says whether you think there are potential applications of NLRs as targets for improving plant resistance, for example, new breeding technologies or manipulation of these proteins, but whether that will have off targets or detrimental effects for the plant too.
I've heard I mean, a long time ago when they started making disease-resistant plants, they did it in a very with technology that they weren't really sure exactly what they were doing. Apologies if that insults anyone, but in the technology it was a bit more crude. Now we can do things much more targeted ways. I don't think that these are proteins that are very low expression level, I don't think it will be a burden to the plant to have them, as long as you have the correct accessory components with it, they should function. In terms of how would we provide disease resistance, there are plenty of different pathogens that form structures like Hostoria, the ones that I mentioned. They're fungi and other oomycete pathogens. We could redirect NLRs to this site and maybe improve disease resistance that way. That's something we're definitely interested in trying in the future.
Okay, so now there's another question for Jack. So the question is, what do you think happens between the chloride efflux and NLRP3 activation? So what exactly triggers the activation in this case? Or how the chloride acts, I guess.
That's a very good question, and one I really want to find out. Yeah, I think like the relationship between them needs to be further investigated. We don't know whether chloride efflux is helping potassium efflux, and there's a range of kinases I want to have a look at, see whether they're also implicated. But I don't have a good answer for that right now. But it is something I really want to look into in more detail.
Okay. And there's another question, I guess it's for Cian. Says, where does the effectors of P. infestans localizes?
Sorry, I missed the
where does the effectors of P. infestans localizes?
Yeah, so they localize to a wide variety of compartments, but what we have noticed is, in particular, the ones that are which NRC4 provides resistance against, mostly localized to the same place as NRC4. So they're in the same place at the same time. So we think they're guilty, and we know that these are proteins. These effector proteins, these virulence factors. We know that their job is to prevent secretion of defense-related compounds. So they prevent the plant from secreting defense-related compounds, and then we think NRC4 and its sensors are trying to sense this and stop it from happening.
Okay. This was coming. So one for Jack. Hi Jack. How does potassium efflux activate? How and does potassium efflux activate the inflammasome? And I guess again it's a speculation of how would they do that.
Yeah. So there's it was reported that potassium efflux caused the activation by promoting the binding of an accessory protein NEC7 to NLRP3. But it's getting a lot more complicated the more the research is going on into this area. So I don't think anybody knows entirely how it's all coordinated right now. But I know there's lots of different research groups are intensely looking at NLRP3 activation, and I don't think it'll be very long until we have these answers. It just seems to elude us all, like the specifics between them.
Mm-hmm. Thank you. And I guess that bringing that question from the plant to the mammals and vice versa is NLRs in the plants also known to be regulated by ion fluxes like the mammalian counterparts, or something that is not known, Cian?
Sorry, every time I get unmuted, somebody speaks in my ear and I can't hear part of your question.
I'm asking whether you know the link between the mammalian and the plant's NLR. Mm-hmm. So we know they're very different in evolution, but do we know if ion fluxes are also regulating the plant's NLRs as they are for the mammalian?
Quite possibly. I think if I understand the mammalian NLRs, it's more of how they get activated initially, right? So plant NLRs probably rely on ion fluxes to actually trigger the cell death. We know that when the cells die, they release ions for sure. That's fairly obvious, but we don't think that they get activated by ion influxes. At least not specifically. It might have a secondary knock-on effect to neighboring cells that will non-specifically activate neighboring cells and tell them, okay, you need to kill yourself as well. So that might happen, but we don't have the technology to dissect that, because whilst each cell in a plant is autonomous, more or less, they're all stuck together. So you can't really do single cell experiments as easily as you can with mammalian stuff.
Okay, and then related to that, you also mentioned that the sensor really doesn't necessarily bind a molecule to sense the pathogen. So what are they sensing then?
So that's a good question. Sometimes they do bind the effector directly. But if you think about it, that's not as good for evolution, because all the pathogen needs to do is change the shape of the effector, and they've lost the resistance. Generally, what they do is they guard very important plant proteins or lipids or whatever, and they wait for any pathogen, doesn't matter what it is, to trip the wire. So they wait until they manipulate something really important, and if they see that changes, they trigger cell death.
Yeah, so I guess this in a similar way to the ion flux, but maybe something different happening. Okay, so I think in the interest of time, we'll leave it there. Well, I think this just the last question. I'm not sure who is directed to. I think maybe both. It says if there's any way to slow down the activity of effectors. So I don't know who feels. I guess it's talking to the ability of sensing that you were talking about, that the effectors might sense it. I'm not sure. I guess you could, if you inhibit the NLRs, you can probably slow down the activity of these, but I'm not sure.
Yeah, I'm not. I mean, in terms of slowing down to prevent them from triggering cell death, some pathogens you can knock them out, but generally, for our pathogen, you can't really do genetics on it, so it's tricky.
Okay. Okay, so then, sorry, Jack, you.
I was just going to say, in terms of NLRP3, there are certain stimuli that seem to have slowed down kinetics themselves, and so they don't undergo pyroptosis. So there are mechanisms to limit the effectors of NLRP3, but again, how that's achieved is still up for debate.
Yeah, that's a big debate. We could start here. How that's achieved. Yeah. Okay, so I'd like to thank you both for great talks. Thank you. Before we go, I'd like to remind you of a couple of things. So I think I will encourage everyone, if you want to follow the conversation online. So just on Twitter, you can follow Biochemical Society and PPP Publishing. So if you have any other questions or any other thing you'd like to comment on the webinar, I think that would be a great place to do that. Of course, also, if you want to suggest future topics that you think will be interesting to a wide variety of people and for the Biochemistry Society, please go into the website and you can submit a proposal. If you're not sure, I guess you can also ask one of me or the organizer to point you in the right direction. And I would like to remind you of the next webinar, and will be the series entitled Digital Biology, Advanced Computational Approaches to Biology Design and Engineering. And this will be a great webinar as well to learn about the uses of advanced computational approaches to examine existing and engineer novel biological processes. And this webinar we'll hear from Professor Christian Berger and Dr. Nicole Piercy to share their research and their discoveries in this field. And finally, I would like to also encourage everyone to join the Biochemical Society if you still not joined yet. It's a great society that, I mean, before COVID times, obviously, they organized really good conferences face-to-face. But now they're trying to compensate, and they're organizing a lot of online events that I think can help us a lot going through these weird time and challenging times. So please go into the website and if you can join the society, because it will be quite good for pushing your careers and learning a lot of new things. So with that, I would like to finish. Thank you. Thank the speakers again and all the people for joining us today. Thank you very much. Bye.